Broadcast. We're go, Madeline.
Okay. Good morning, everyone. We're going to give people a few minutes to hop into this webinar, and then we'll get started.
Okay.
And you ready to go YouTube Live?
YouTube is preparing to stream live to the webinar, so we should be live in about 4 or 5 seconds here.
Awesome. Well, good morning, everyone. My name is Madeline DeLapp. I am the Operations Director for Humboldt County Growers Alliance. HCGA is the trade association representing licensed cannabis.
Is preparing to stream live to
That happened yesterday. HCGA is the trade association representing licensed cannabis businesses in Humboldt County. And we are pleased to bring you a week of education at our Cannabis Genetics and Bioethics Conference, which is produced in partnership with Hendricks Farm. And specifically, it's been a pleasure to work with my colleague, Daniel Hendricks, who is on the panel today. Every day this week at 11 a.m., we are bringing and have been bringing different tools of the trade. On Monday, we talked about microfarming. On Tuesday, viruses, viroids, and pathology. On Wednesday, we spoke about tissue culture and meristematic restoration in cannabis. Today, we are talking about gene editing for yield and stress tolerance. And on Friday, we are discussing genetic storage for common access. At 1 p.m. on Friday, we are bringing the Cannabis Breeders' Rights and Bioethics panel with Nat Pennington of Humboldt Seed Company, Jerry Whitting, LeBlanc CNE, Jesse Dodd, BioVortex, Sunshine Johnston from Sunbolt Grown, Eleanor Kuntz from Leafworks DNA, and this panel is going to be moderated by Colleen Byers from Breeders' Best. And what they're going to be discussing are all of the practices and applications brought forward over this week and that are available to cannabis breeders and small farmers. All of these panels need to be registered for independently. So if you have not registered for Friday's events, make sure you do so on hcga.co/Cannabis Genetics and Bioethics Conference. And if you missed any of the panels on either Monday, Tuesday, or Wednesday, we've gotten those posted to our website, hcga.co, or you can check them out on Hendricks Farm's YouTube channel. And with that, I'd like to introduce my colleague, Heather Luther, to talk about how we are able to bring to you this free educational programming.
Thanks, Madeline. Good morning, everyone. So usually you'd be able to meet some of our sponsors by wandering around a B2B conference in person, but since we're not doing that today, I'm going to read a little bit about some of our sponsors, and we really appreciate them being able to bring this to you for free. So our sponsors for this whole week have been Tag Risk Insurance, Rocky Mountain Cannabis Consulting, Volks Amendments, the Humboldt County Business Development Center through a grant from Headwaters Fund, and today's sponsor is RadSource Technology. So I'm going to share my screen here for a second and share a little presentation for you guys. Can you all see that? Okay, so RadSource Technology is actually, they come from the medical industry, and they're manufacturers of photonic decontamination solutions for a wide variety of applications, including blood transfusions, cancer and tissue research, sterile insect technology, small animal research, food, and more. They discovered that the same clean technology they're using in the medical industry can actually be used to protect, preserve, and sustain life for cannabis. So they provide 99.9% decontamination of microbes on cannabis. Over here you can see some of the benefits. It helps remediate mold, mildew, and other microbials. I'm going to scroll down to the next, and I'm going to share this for those of you who are on our Slack. I'm going to share this if you want to learn more. Their goal by being a member with Humboldt County Growers Alliance is to participate, educate, and support HCGA growers on making the most profit per crop. Their mission is to make sure cultivators don't lose flower and profits due to failed state microbe testing, which is a real concern, as we all know. Some of the benefits to their decontamination is that it's a cold technology, so it doesn't heat the flower, which can create premature decarboxylation of THCA, which is not something we want. Sorry, I'm having a cat fight in the back here. It's also chemical and residual free, so it doesn't saturate the flower with any disinfectants that people don't want to smoke or leave potential residuals. It's safe, clean, and green. It actually utilizes short wavelength electromagnetic energy, which is really cool. So this technology is not found anywhere else, and it's very reliable. You can see some examples of their equipment over here. If you want to learn more about this, here is Jeff's contact. You can call him at 310-460-6050 or email him at jduvall@radsource.com. That's JDUVALL@radsource.com. There's a lot of companies out there doing decontamination, and they want to tell you not to be fooled by false decontamination claims. So learn more with them and you can pass more tests. So thank you so much, RadSource Technology.
Awesome. Thank you, Heather, and thank you, RadSource. HCGA is an organization that is supported by its members, including both our licensed cannabis businesses as well as our allied industry businesses. And we really appreciate the support that allows us to continue our mission. And with that, I'd like to introduce Daniel Hendricks. Daniel is from Hendricks Farm, and I've had the honor of working with him for the past several months as he has produced this series of panels and bringing forward the educational programming that we're presenting this week. Daniel, would you like to introduce this panel?
Hey, everybody. Yep, thanks for coming. This is a really important panel. We kind of went through, I said it before, went all around last year to mini science conferences, some specific to cannabis, some not, and you know, brought back some of the most interesting technology and tried to consolidate to have this conference where we could share the information that we learned. And you know, we met Venki Pegadaraju in Las Vegas at the Indoor Ag Conference, and his presentation about a specific gene that he discovered in many crops, one of them including cannabis, was just really powerful. And he was nice enough to put this presentation together specific to cannabis, which I'm really grateful for. And then, of course, Eleanor Kuntz, we have a great relationship with her. She's done a lot of work with Candor and cannabis sample herbarium, as well as Leafworks, which is cannabis gene sequencing and you know, gender identification business. And she is the founder of that. Both great people, and we're going to hand it over to Eleanor to kind of give us an introduction into gene modification and gene editing, and then to Venki to share his technology. Thank you. Go ahead, Eleanor, you're on. Yeah,
Great. Well, it's a pleasure to be here. It's always exciting to get to talk science with a bunch of passionate plant people. And I just put together a little basic definition so that we're all on the same page, because when we speak of GMO and gene editing, there are a lot of different opinions about what is GMO. And so the first thing we're going to do is start with some definitions. So one of the issues in GMO is just that there is deeply problematic because there's not a commonly held definition. So the most basic definition of GMO is any organism that's altered in any way that does not occur in nature by mating or natural recombination. And so when we read this definition, you should be very surprised by the fact that all selective breeding, whether it's in vitro fertilization, the utility of sperm banks, cloning, etc., under this definition, pegs those organisms as genetically modified. So the European Union has a pretty broad definition of what is a GMO. So they define GMO as anything that is generated by selective breeding and other means of artificial selection. So they've later reeled back that definition slightly so that it specifically excluded traditional breeding, in vitro fertilization, the usage of polyploidy, mutagenesis, and various cell fusion techniques that we use in classic breeding schemes. So that anything that doesn't use recombinant nucleic acids or genetically modified bits of DNA in the organism. The USDA has a very different definition of GMO from the EU. So with the USDA, they consider GMOs to be plants or animals with heritable changes introduced specifically by genetic engineering, so much narrower definition. And then they consider GEO, so genetically engineered organisms, to specifically refer to organisms that have genes that are introduced, eliminated, or rearranged using molecular biology, and particularly recombinant DNA technologies such as transgenesis. And so the USDA has ruled that gene-edited organisms are not considered GMOs. So this is something that's very different in the US landscape as compared to the European landscape. So as we go through these definitions, I think we can see what the problem is that the definition of what is a GMO, what is a GEO, is really focused on the process more than the product. So this results in a situation where there are GMOs and non-GMOs that have very similar genotypes and phenotypes, and it can be very difficult to tell those things apart. So what can be done with genetic modification? So in general, we can add new genes into organisms from other outside species. We can mess around or change the endogenous or genes that are held within that organism's genome. Genes can be specifically enhanced, alternately expressed, so turned up or turned down in their expression levels, or knocked out, meaning made to be useless so that they're no longer functional. So these genes can be moved within the same species or across different species, and in fact can be moved across kingdoms. So you can have very wide movement between genes into organisms. And the production of genetically modified organisms is a complicated multi-step process. And for targeted changes, you need to know a lot about the genes that are being moved and the recipient genome. So that's something that's very important to keep in mind. And with all of the techniques that fall under the genetic modification umbrella, originally these were all random insertions, mostly using viral and bacterial vectors, occasionally using mutagenesis. But as we've evolved in the GMO landscape, most approaches these days are exceptionally targeted. And so they utilize the double strand breaks that naturally occur in organisms, and they leverage those molecules that fix those systems once they break, so the homologous recombination repair systems that are native in organisms, to insert novel DNA. So these insertions move genetic material into exact locations, which is exceptionally powerful. So gene editing is a little bit different in that you're using either meganucleases, zinc finger nucleases, transcription activator-like effectors, which are TALENs, or the Cas9 guided system, which is CRISPR, which we've heard a lot about, to move these pieces of DNA around. In general, TALEN has really specific insertion capabilities, and CRISPR has become so fashionable because it's quite easy to design and implement changes. So just a brief history of production. So if you think about it in the most abstract way, humans have been modifying plants and animals since the first domesticated plants, roughly 12,000 years ago. But the first genetically modified organisms that we would probably think of happened in the early 70s with bacteria that were resistant to kanamycin. And then quickly we moved into the production of genetically engineered animals in 74. In 82, the first plant was tobacco using Agribacterium, and this should say 1982. We also had the first genetically modified crop which was tobacco for antibiotic resistance. And then the first commercially available food was the Flavor Saver tomato in 1984. So we've had a very long history of genetically modifying organisms, but have rapidly moved into the specific engineering of plants in the 80s. So with that brief history, I'm gonna give it over to Dr. Venki to give us some insight into what he's specifically doing in this arena, and I will be monitoring the chat section for questions, and we'll direct questions to Venki as they come up. So thank you.
All right. Thank you, Eleanor. I'm just trying to share my screen here. Just give me 1 second. Sure. All right. Can everybody see my screen here? Okay.
Yes.
All right, good. So good morning, everybody, and really excited to be a part of this virtual conference. I want to thank Daniel and Eleanor for giving Agribody an opportunity to discuss the applications of our technology in the context of cannabis breeding. So I want to kick off my presentation with this quote from George Washington that agriculture is the most healthful, most useful, and the most noble employment of men. And he mentioned this roughly about 227 years ago, and since then I've made a slight modification by adding woman in there. So this quote couldn't be more relevant under our current days, scenarios of where whole of the world is trying to fight COVID-19 by practicing social distancing and taking shelter in place, whereas our farming communities are really working hard to ensure food security for all of us. So I really salute all our farmers and the farming communities globally for doing such a noble job. Slight change in the title of my presentation. Earlier it was something like the DNA of cannabis. I promise we are going to talk about genome editing, but I wanted to bring a little bit more focus into the scope of our discussion by describing our proprietary, genetic switch. It's a universal genetic switch present in all the plant species, including cannabis. And so I'm going to describe how you can use this to improve yield and stress tolerance in cannabis. Quick peek at the agenda here today, the historic perspective on plant breeding. Although Eleanor touched upon this, I wanted to expand a little bit on that. The idea here is that, what we can learn, sort of draw some parallels from other crop improvement programs and how breeders have exploited the modern breeding tools to make incremental changes, and the idea is how we can use that to readily adapt it for cannabis breeding program. Then compare and contrast various crop improvement strategies, the features, benefits, and the regulatory governance around genome editing and GMOs and conventional breeding. I want to go a little bit deeper into this, and then talk about our proprietary gene switch, and I will describe our technology here and also use some specific use cases to demonstrate how we have successfully used this technology to improve yield and stress tolerance in variety of crops, and then wrap it up with business models for engagement with our clients. So here, it's kind of a historic perspective on plant breeding. We as human beings have been practicing plant breeding since the dawn of the civilization, and shown here it's a kind of a time scale and some relative discoveries that have happened time and again, and that literally has transformed agriculture and given powerful tools to breeders to make incremental changes in the breeding program. So, for instance, if you look at a crop like corn, prior to 1930s, it was mostly open pollinating varieties where there was hardly a yield of about 30 bushel acre or so. And subsequent to that, with the advent of double cross and single cross hybrids, were able to take the yield to about 100 bushel an acre. Moving forward with the advent of molecular tools, as Eleanor mentioned, that with GMOs and also with molecular breeding, we're able to increase the yield of corn about 200 bushel an acre. So these are significant achievements, that could be attributed, directly to the technological developments. And so in the future, the incremental growth would come from, modern technologies like genome editing or through a combination of genome editing and molecular breeding. I just wanted to kind of use this slide here to describe how powerful the modern tools or, like genome editing could be. And by using this very specific example, and I've used tomato here in this case, you can see on the top, is a figure that shows the evolution of tomato from the wild relative, which is on the extreme left-hand side. You can see Solanum tympanifolium, and then you have an intermediate type, and then all the way on the right, you can see the domesticated tomato, which is the modern day tomato, bigger in size and more aroma and flavors. And so this actually time scale is roughly around 80,000 years as per the best estimates. And below shown is kind of a de novo domestication of tomato using a genome editing strategy, which took like, maybe a few generations. And essentially, because there's a lot of genomic research that has happened in tomato, and we have a lot of sequencing data on wild and land races and domestic tomatoes. So the researchers in this case, were able to identify the genes that were important for the domestication of tomatoes. And they went back and genome edited those specific genes in the wild progenitor lines, and they were able to create a version of tomato that's closer to the domestication version. And so this, you can see this, the power of this this technology, that within few generations, you're able to convert a wild into somewhat domestic, closer to domestic. So essentially, genome editing is a very powerful tool in the hands of plant breeders to accelerate, what has been happening in the nature. We're able to do that in the lab to ensure that breeders can rapidly release new varieties to meet the market needs. So what is genome editing? So genome editing or genome editing is a set of tools that researchers use to make changes within the DNA of the cell. So you can either add or delete or replace a certain segment of the DNA within the cell to change the function of the gene. And so one of the key components, as Eleanor has touched upon in the genome editing, are the nucleases. And so mentioned here a range of nucleases, and I won't have time to go into details and all of these, but it's I think it would be suffice to say that all of them are proven important. But one of the things that has become really popular nuclease is the CRISPR-Cas system, and so CRISPR has become really popular because it's fast, it's cheaper, it's efficient, and it's accurate. So the complex that's shown here, the CRISPR-Cas9 complex, consists of the guide RNA and then the Cas9, which is attached to it. And so the function of the guide RNA is basically to cruise through the genome and find its target and where you intend to make the change. And subsequent to that, the Cas9 kind of creates a double-stranded nick. Once this happens, the researchers, they use a cell's natural repair mechanism either to add or delete a piece of DNA, and that process is known as non-homologous end joining method. Or you can replace the existing segment of the DNA with more of a customized piece of the DNA, which is known as the homology directed repair mechanism. So what I've tried to show here is, various crop improvement strategies. On the top, you can see conventional, middle is the transgenic, and then the genome editing. And I try to kind of compare and contrast, several features with this. In terms of the R&D timeline, if you look at the conventional breeding process, it's very laborious, and it can take anywhere from 10 to 15 years, depending on the crop that you're working on. Although you may have a chance to expedite that when you use molecular markers. Transgenics, on the other hand, can take anywhere from 3 to 5 years, whereas genome editing is about a year or less, depending on the species. In terms of regulations, conventional breeding or mutational breedings, both of them, since they do not involve any transfer of foreign DNA, so they don't need to be regulated. Similarly, in case of genome editing, if you make a small deletion, you don't have to regulate those. Whereas, since transgenics involve insertion of the foreign DNA, they have to be regulated under the Article of 7 CFR Part 240. Similarly, labeling, no labeling requirement for conventional as well as for genoming, whereas there is a public law that mandates you to label the GMO crops. In terms of the cost structure, it's standard. Like you have a variety that is you want to license from a breeding company. You can license it and then you can pay royalties. Whereas transgenic and genome editing have a similar cost structure, which involves tech access fees, milestones payments, and also royalties. How are these crops regulated? So here it's a very simplistic view shown here on one end. On the left-hand side, you have the conventional breeding, and on the extreme right, you have the GMO, and in the middle is the genome editing. Specifically the technologies that we are developing at Agribody fit into this SBN1 category. Which means you're either likely regulated or not regulated depending on the jurisdiction where you market these products, as opposed to the GMOs, which require millions of dollars for deregulating, and then even then you're not sure whether your product will make it to the marketplace. So this is a streamlined process under the USDA. It's called the AMI regulated governing body. And if you're a researcher that's working in the genome editing field and not sure whether your product meets regulation or does not need any regulation, you submit a letter of inquiry to USDA and they will review, and you disclose exactly what has gone into to develop the product, and then based on that, USDA will basically say either regulated or you're not regulated. And this is a slightly outdated slide. Last time I checked, there were about 90 letters that were submitted. In most cases, it was favorably viewed, and they they approved for release of those products. Some examples of products that have gone through the AMI regulated process, as shown here are 3 different examples. On the extreme right hand side, you have the high oleic acid soybean that was done by Calyx, and they used the talents, and then they did editing on the FAD, fatty acid desaturase gene by creating minor deletion. And then in the middle, you have the non-stem tomatoes from University of Florida, Harry Klee's lab, where they did a CRISPR-Cas method to delete a portion of the J2 gene. And the idea here is that, the stems would not basically be sticking to the fruit, and it will not be stabbing its neighboring tomatoes when it's being transported. So it increases the yield of tomato. And all the way to the right is the Waxy corn from Corteva, which was originally a discovery of a mutation that happened in 1920, and so Corteva emulated this kind of a Waxy mutation, and then they use CRISPR-Cas9 to create a deletion to increase the starch content in the kernels as opposed to the expensive sugars. So it's mostly for industrial applications like glues and so on and so forth. As you can see, I've given the first prize to the high oleic soybean product from Calyx because it is already being commercialized and it's actually being sold in the marketplace right now. So as far as I can remember, this is the first genome edited product to hit the marketplace. So where can we sell our products? So this is a rapidly evolving landscape, in terms of regulations. US, Canada, and some of the South American countries are in favor of genome editing. So either you have to likely or you don't have to regulate genome edited products in these countries. Israel will allow genome editing similar to Australia. And also Japan last year, they came on board, and they are still trying to work out on the regulatory details, but then, it looks very favorable there. So I think roughly around 10 different countries where we can actually sell our products today if you have genome edited products. I want to shift gears a little bit and talk about the genome editing requirements. So what do you need when you are a researcher working in this field? The first and foremost thing is to have a complete genome sequence, a detailed database of resequencing data and full genome sequence data for your crop species, so that you can do a good job for designing and avoid off-target effects. Then you need to have access to validated targets. This is where, Agribody, company like Agribody could play a significant role. So I'm going to describe to you a little bit more about what I mean by that in terms of access to validated targets in my presentation. And then more importantly, a delivery mechanism of tissue culture and transformation where you can reliably introduce your DNA and at the same time regenerate your benefits from the tissue culture process. So our big idea, for innovative seed companies and also for cannabis growers that are dissatisfied with yield and disease resistance is that, through our patented gene switch or genome editing technology, we can increase the yields by about 20%. At the same time, confer stress tolerance or disease resistance to the plants. And the way we do that is by doing a small unregulated deletion in a gene that's involved in determining the sulfate specificity. And when I mean unregulated, I'm actually referring to the governmental regulation process. And importantly, we have proven this technology both under greenhouse as well as in the field trial conditions. The biggest advantage of doing this via genome editing as opposed to GMO is that you can avoid all the social pushbacks and the cost and the time that associated with creating a product. It's much faster in case of genome editing. So here's the core of our technology. Think of this as a switch, and so there are two components in there. One is the EAF5A, which stands for eukaryotic initiation factor 5A. And then DHS, which encodes deoxyhyposin synthase. So as the name suggests, the function of this is to hypusynolate the EAF5A. DHS post-translationally decorates the EAF5A gene by adding a spermidine molecule to the conserved lysine residue and turns it into an hyposylated form. So this particular switch is, like I said, is very conserved in all the plants, animals, and all the 5 of the 6 living kingdoms. And in all these cells, what happens is that, the cells, they sense the ratio of this, the native form, which is a lysine form, and the active form, which is a hyposylated form. And then the cells make a judgment about, whether they should grow or continue to divide, or they should progress towards the senescence mechanisms. So several years ago, the co-founder of the company, John Thompson, showed that under plants that are senescing, there is an increased expression of DHS and there's an increased accumulation of DHS, protein accumulation of DHS. And so John did exactly what any biotechnologist would do. He did the opposite of what nature intends, and so that's what most of the biotechnologists do. And so he downregulated the expression of a DHS. And as a result of which, you have a, reduced accumulation of the hyposylated variety, or alternatively, you can actually increase the expression of, increase the expression of the native form of this. In both the cases, it resulted in the triggering of the switch, which led to very interesting phenotypes in the plants, like extended shelf life of perishable products, increased biomass and seed yield, and at the same time, you're able to get enhanced tolerance to stress and diseases. Now, it turns out that life is a little bit more complicated than a simple cartoon switch, and that EAF5A comes in different flavors. Shown here are 3 different isoforms, but in some of the crop species, it could be even 4 or 5. So what we have done through extensive research is to, functionally characterize each of these isotypes, and we have shown that EIF5A is the senescence isoform. So if you downregulate the expression of this gene, you can get extended shelf life in plants. And similarly, the EF5A2 is a pathogen isoform. And if you downregulate this gene, you can get enhanced disease resistance. And similarly, when you upregulate EF5A gene, you can get enhanced seed yield and biomass. What's interesting is that, just by downregulating DHS gene alone, you can actually uncover all these positive traits. The thing to remember here is that the switch that I mentioned is actually an analog switch as opposed to a digital switch, which means that you have to carefully modulate the expression of this gene. So you can't completely shut off, or you can't, you can't basically complete all these genes. So there's a sweet spot where you can, you have to modulate this expression so that you can get all these interesting phenotypes in plants. So here is some data here I will show you with starting off with canola plant here by downregulating DHS. John showed that, there is an increase of about 50 to 300% increase in vegetative as well as in the reproductive mass of the plants. And you can see here on the left-hand side, control versus the DHS downregulated plant, there is an enhanced leaf surface area, and then also increased root architecture, the structure, more enlarged roots, and which in turn was, associated with, providing the stress tolerance in the plants against a range of stresses like drought, nutrient, salt, heat, and cold stress. So I showed increased leaf surface area, and then John also measured that these plants with downregulated DHS expression was also associated with increased photosynthesis, more green in color, and which in turn translated into an increased yield in the plants. And you can see compared to control plants, there was an increase in yield by about threefold in the DHS regulated plants. Similarly, in case of tomato, downregulation of DHS, increased the growth of the plants in both the cases that are shown here. On the left-hand side, you can see the tomato control versus the DHS plants doing much more better, more robust growth. Similarly, in case of the banana, controlled, and the DHS downregulated banana are doing much better. So not only that, but the company which did this project, it was an Israeli company called Ram Meristem. Actually, they took it to the field trials, and they also measured some of the other interesting phenotypes. And shown here is the disease-resistant phenotype. On the left-hand side, you can see the control plants infected with Sigatoka, which is a necrotrophic fungus, and you can see a clear manifestation of the disease, as shown here by this yellow arrow here. As opposed to that, there is the Agribody DHS downregulated banana, literally don't have any symptoms. So I'll be quite honest here, and this is actually more of a tolerance mechanism than a complete resistance. So the company that did this project, although they were excited to see these interesting phenotypes, they decided not to commercialize at the moment because it was not worth their time or efforts to take a product that's offering partial resistance. So right now they're kind of stacking up with other resistance genes in order to get more durable resistance. So you can, instead of having a single gene, if you have multiple genes for resistance, you can get much more enhanced durability in the plants. And similarly, on the right hand side, you can see the tomato plants, compared to the controls, the EI5A2 in this case, downregulated tomato plants are much better and more tolerant to the bacterial infection. Another interesting phenotype that they notice is in terms of shelf life extension. So shown here on the top are the tomato controls and the Agribody on the below, and you can see that the 6-week-old tomato is much better than the 2-week-old control plants, and there is clearly a spoilage and softening of the tissue happening there. One of the interesting things I want to mention here is that although people in the past have tried, several approaches for increasing the shelf life, although they were successful in increasing the shelf life, but they compromised on the aroma and other quality aspects of the fruit, and which wasn't really that successful. But in our case, since our gene is in the upstream senescence pathway, it does not really mess around with any of the downstream biochemistry. So, as a result of which you're increasing shelf life and at the same time ensuring that all your flavor and aroma characteristics are retained within the plant. Similarly, on the bottom, you can see in case of bananas, control plants, clearly perished at the day 14, whereas Agribody DHS downregulated plants are doing, much better. So it's essentially, a threefold increase in the shelf life in these perishable produce. And also an interesting thing we observed in case of carnations, when we downregulated DHS, it increased the shelf life by about threefold. Very clearly, you can see this at day 21 that the control is a complete close, whereas the DHS downregulated carnations looks very fresh. So increased shelf life of carnations, really good, but we want to really demonstrate this in a useful crop like cut roses, and so which is exactly what we are doing at the moment. We have a partnership with a local startup here in town that is actually trying to downregulate DHS gene to increase shelf lives in roses. Then I want to, I want to kind of share with you some exciting field trial data in alfalfa. But before that, I want to share a little statistics here in terms of alfalfa. Is the third most valuable crop in US next to corn and soybean. It's an $8 billion market. About 1 billion is in exports to countries like, Japan, China, and Middle East. And the interesting thing about the alfalfa breeding is that it has not seen any yield improvement in the last 30 years, or at least directly. So what we did about 6 to 7 years ago, and this was the work that was done in collaboration with Alan Bennett's lab, that we overexpressed one of the genes of the EF5A3 in alfalfa, and we showed that under 2 years of consecutive field trials, there was an increase of about 20 to 45% yield enhancement in the alfalfa. And if you are familiar with alfalfa breeding, the challenge in alfalfa breeding is every time you try to increase the yield, there's a compromise in the quality of the forage, and it doesn't, it's not a useful product for the feedstock. Whereas in this case, what we noticed, interestingly, that despite a significant increase in the yield, the quality remained the same. So there was no change in the quality. And so this, there's a detailed manuscript that's available. If anyone of you is interested, I'm happy to share more details about the field trials, how it was done, and other details of this particular project. So a little bit of information about the DHS. We know a lot about this DHS gene in several crops, and shown here is a gene structure of 33 different DHS genes in 30 different species. There are several crops here: monocots, dicots, perennials. You can see the genus and the species on the right-hand side. On the left-hand side, you see the detailed gene structure. What you can see here are the gray boxes, which are the exons. And the thin line is the intron there. I would like to point you specifically to these three segments here. One is a pink bar, which is the active site of DHS, and then there is this red bar here on the fourth exon. As you can see, that's the K144 site. And at least if the in vitro human studies, we have shown that a mutation in this particular residue here does not alter the spermidine transfer activity, but then it kind of significantly reduces the NADH hydrolysis step by about 80%. So it's a very interesting target, as we are trying to follow this in some of the crops, particularly that ones are the ones that require kind of a hybrid makeup. And similarly, on the second exon that we have also identified several mutations, inframe mutations that this gene can tolerate, and could be a great candidates for us. So essentially, we know a lot about these genes, and we have proven that by reducing the activity of this gene through genome editing, we can recapitulate all the interesting phenotypes that we are able to document with the antisense DHS downregulated. So I want to show you one specific example here in case of rice, and this was actually a patent application that was submitted last summer. And so there's some data that went in to support the patent in two different crops, one monocot and one dicot. And this is on the left-hand side, you can see the rice plants, the wild type compared to the genome edited rice, where essentially we deleted a 10 base pair region within one copy of the DHS. So it's actually a monoallelic mutation. And in the heterozygous state, you can see that plants look, similar architecture, whereas, compared to the control plants, the rice plants that are genome edited are much more greener in color. There is no difference in the panicle numbers. But then if you look at very carefully on the grain, there is a 20% increase in the size of the rice grain. And similarly, on the bottom, you can see the canola plants. These are coming straight off the greenhouse, and we are trying to further characterize these. But, very preliminary evidence looks very promising that the leaves are much more expanded and they're much more greener, and which is what we noticed earlier in case of DHS downregulated plants, similar sort of phenotypes. So we are very excited about, characterizing this further. So we have very unusual as a startup in the sense that, we have about 15 issued US patents, several foreign counterparts, and then we also have the ability to sub-license the Benson Health's CRISPR 3.0 nuclease. So in a sense, you know, we are a one-stop shop. So if you partner with us, you know, you get access to validated targets that we have shown in more than a dozen of crops work really well. And at the same time, we have the ability to sub-license the CRISPR tools that you need to use it for, you know, in your breeding program. So yeah, so pretty broad claims and, you know, a strong patent portfolio. So at this point of time, you know, anybody who messes with EF5A or DHS would be stepping on our patents. So real quick slide here on the business model. So essentially, we have, you know, at least 2 functional working models here. The third one we hope to be able to execute in the future. But predominantly right now, all of our projects, majority of the projects are the licensing projects, which, you know, involve typically a tech access fee, benchmark payments, and royalties. In some cases, you know, we have done co-development projects. It's mostly for scenarios where people don't have laboratories and don't have infrastructure, so we partner with them, and then you know, then reward it for the net trade value as opposed to royalty payments. So these are the different crops. Examples of, you know, the licensing crops that we have done deals with in potato, coffee, camelina, rose, and lettuce. We have 2 co-development projects going, one in alfalfa, another one tomato. And in future, like I said, you know, this is what we are working towards, is to get an integration model in place. So the idea here is that, you know, Agribody will invest in creating certain donor lines, and then we will basically partner with companies to transfer our DHS genome edited genes into, you know, the elite lines of the seed companies. So this, the real advantage of doing this is that, you know, it'll cut down the genome editing cost, which is still higher. And then we'll be able to rapidly develop products to the marketplace, and we'll be selling a product as opposed to just an IP. And so that way, you know, that gives us an ability to create a brand identity for ourselves. So this is a quick peek at our team. You know, Jerry Feidelson, he's the co-founder, CEO of the company, and John Thompson was the founder of the company, and myself, I joined a couple of years ago with Agribody, and then we also have some excellent financial as well as legal counsel in our team. We also have a stellar team of consultants and advisors. And a lot of the work that I showed you in ALFALFA was done in collaboration with Alan Bennett's lab in UC Davis, and several other you know, serial entrepreneurs that are on board. So a quick attraction here on slide, you know, what are the projects that we have done so far in the list of companies here. You can see GR Simplot, and we are particularly excited about this because recently, you know, GR Simplot shared about the yield trial results in potatoes, and which they showed that in the second year of the field trials, they have seen some significant improvements in the yield of Russet Burbank, and they are now committed to doing a third year expanded field trials in additional locations. So we are pretty excited about this project. We also have a project going on with a South African company called Tomatoes to Genome Edit Tomatoes to increase shelf life. We have a project going on in coffee, alfalfa, 3-week roses that I mentioned, and also you know, a couple of projects in, you know, camelina as well as in lettuce. So essentially, to summarize, you know, we have a proven technology. We have a lot of data that we have shown in the last several years, very broad IP and field trial data. We have an experienced team, you know, great resources, and full serial entrepreneurs in our team. And most importantly, we are seeing some great traction in the marketplace. So I invite all of you to join Agribody either as a client or as an investor in investing in this such novel technology. So with that, I will kind of stop here, and I'll be happy to take any questions or comments on this technology. Thank you very much.
So I have a few questions in the question and answer. The most important one, maybe to all of us, is that they look good and the yield is better. However, have you tasted and compared the quality of the plants? Do they have the same or different amount of nutrients? And then again, just about the taste and the smell of these products.
Talking about alfalfa in particular?
Or with the tomatoes, either.
I'm sure there's some work that has been done there. I don't have access to the data yet, but the manuscript that I mentioned, happy to share, where we clearly showed that in alfalfa, despite the increase in yield, none of the quality characters, like we measured, the biochemical characteristics in terms of the fiber quality and everything remained the same, so they didn't change at all.
Excellent. And then we have another tough question. Are there any potentially detrimental consequences to genome editing? For example, what happens if a genome edited population escapes and cross-pollinates with wild populations or neighboring heirloom varieties?
Yeah, so that's a risk which much similarly what we have experienced in case of transgenics as well, right? And I don't see any different than that in case of genome editing. And so you have to be really careful about those things in terms of understand the crop flowering mechanisms, and and then make sure that you have perfect borders and everything to protect. So the similar sort of rules would apply there as well. And also it depends on what kind of choices you make about traits, right? So you gotta be a little careful there.
Yeah, I would add on to this. This is one of the things that I spent an extensive amount of time studying, and there's no easy answer to this question. It really does depend on the specific types of changes that are made to those organisms and how selectively advantageous those changes are in the wild, right? So you can imagine if you're making a change that offers some huge advantage, that it would sweep through the population and could potentially have a very damaging effect on nearby wild or heirloom varieties that can cross with the genome edited plant. But if you have something that doesn't have a specific selective advantage, it's quite difficult to move novel genes into a large population. So it really depends on what's going on, just like Dr. Venki said. So complicated. Plants, organisms are complicated, so there's no easy answer to that question.
Sure.
And then we have one more specific question. What effects have you seen on tomato trichome production?
We haven't gone that detail, and so let me back up a little bit. So this was a work done by John Thompson, and he did this about 6 or 7 years ago, where he downregulated some of his gene using, and that was like through antisense approach, right? And and he witnessed all these interesting phenotypes. But right now we have a project going on with UC Davis where we are trying to genome edit tomatoes for the DHS gene. And so this is where it becomes really interesting, because this is the product which we aren't actually commercialize. And so this is where we are going to give a lot of emphasis on detailed phenotypic characterizations of all the traits that you mentioned. So, but to answer your question, we haven't looked at the trichome development, but it's something that something to watch for. So we'll keep an eye for that.
Great. We have another really great question about heritability. And so, are these gene edits always heritable? And what would the effect of edits do if they were bred with non-edited plants? Would the edits be discoverable through sequencing in the heterozygote and therefore still subject to intellectual property laws, or would the edits fade away?
So very
I guess,
interesting
heritability
question as well.
Yeah, very complicated.
So in terms of heritability, yeah, it is heritable. I mean, our integration model is basically based on that assumption that we create donor lines and then we integrate our target mutation into other elite lines. So it is heritable from that standpoint, and it's because you ensure that you make enough of effort of characterizing these plants in greenhouse as well as in field trials, and ensure that it's stable. But there are chances where depending on there be epigenetic changes, or if it goes through the tissue culture process, and if that could induce some of those changes, and there is some element of risk, but that's a very low frequency event. But in general, it is inherited and transmittable like any genetic Mendelian trait.
Great. And there's another question about diseases. You speak about enhanced disease tolerance. So what specific diseases are you seeing enhanced disease resistance or tolerance to?
So in the case of banana, and I showed you is a Sigatoka, it's a necrotrophic fungi. And what's interesting, we know a little bit more about the mode of action of how this works, is that in case of necrotrophic fungi, they like to feed on dead tissues, right? And so they induce the expression of EF5A2 in the plant, and as a result of which, there's a senescence that happens, and then it kills the plant, and the fungi is very happy feeding on the soup of these dead cells. But our strategy was to knock down or downregulate the EF5A expression. That means that you're delaying the senescence process, and as a result of which there's no manifestation of the cell death happening, and then you're getting increased tolerance to the necrotrophic reaction. So that's something that we have shown in case of fungal. On the other hand, in case of bacterial infection like Pseudomonas syringae in tomato, we saw an enhanced tolerance to this bacteria. So those are the two cases, and it kind of, I'm sure will be applicable for, fungus in that category that are necrotrophic in nature, as well as in bacteria that, that involve bacterial secretory process for infection.
Great. And I guess just to speak specifically to this audience, have you identified any specific traits that could be advantageous to cannabis with the technologies that you're currently using?
So our gene switch essentially can give, 3 or maybe 4 important traits. One is the yield enhancement, and I'm sure that will be of a significant value to increasing the number of buds. Or I'm not a cannabis expert, so you can help me in here. So yield for hemp as well as for cannabis, this could be a significant, sort of a trait. And also for stress tolerance, drought tolerance and disease resistance, both abiotic and biotic stresses, this could be a great. For example, I hear that in case of cannabis, they have big issues with downy mildew resistance, downy mildew issues. So this could be a great target for increasing downy mildew resistance. And also the shelf life aspect, it needs to be determined, but I heard from some of them that this could be useful in some cases, particularly if you're trying to use it for blending it with coffee and other things, so depending, I think 2 for sure, but shelf life also in some cases could be a useful trait.
Great. Just scrolling through here, the questions. There's a specific question about haploid induction. Does your company offer haploid induction to protect specific cannabis genetics from others breeding with them?
At the moment, no, and it's because of the model which we have actually embarked on as a startup company, because we are limited on resources. And our idea here is that we are a 2-trick pony. We have 2 genes that are known to work in a range of crops, right? And as opposed to multiple genes working on a single crop. So the model here is that we outsource most of this work to labs that specialize in these transformation methodologies, because like I said, these are 2 genes that work on a range of crops. So if we were to set up an internal facility, it will be a huge undertaking for us. So rather than that, we just wanted to have a more lean operation here, like by kind of outsourcing this to several laboratories. So we don't do any transformation or tissue culture at Agribody. All we do at the moment is by providing licensing and then support at the research and development phase. We contract with our clients and we help them as a consultants and designing of guide RNAs and vector constructions and all the great details, and sometimes even help in the tissue culture process and trying to optimize protocols. We provide our input there, but as such, we do not have laboratories at the moment.
Great. Well, this has been super informative. Thank you so much for all your insights.
Great. Thank you very much. And if you have any additional questions or you want to know more about any of the aspects, feel free to reach out to me at Venki@agribody.com or call me at this number here. So thank you once again, Daniel and Eleanor, for this wonderful opportunity.
Yes, thank you, Venki and Eleanor, and definitely to Daniel. We've got another couple questions that are coming in, so I just want to make sure that we got everything before we wrap up. Just as a reminder, you can check out this, any part of this that you missed on Hendrix's YouTube channel. We're posting this as well to hcga.co under panel recordings. And don't forget to sign up for Friday's panel discussions. Okay, thank you so much, everyone.